So ever picked a bacterial colony and wonder, does this colony actually contain my insert? That's exactly where Colony PCR comes in. Colony PCR is a quick screening method that helps us identify positive clones without doing a plasmid isolation for every single colony. First, look for well-isolated colonies on your agar plate. Now, using a sterile tip, gently touch just a tiny amount of a colony. Remember, less is more. Then transfer those cells into a PCR tube that contains your master mix. Now run PCR using primers that are particularly designed for your insert. Then after amplification, load the PCR products onto an agarose gel and check the bands. Now this is where the real work begins. Now let's look at this hypothetical example. First, check your controls. Basically, a positive control contains DNA that you already know should amplify. So it should produce a band of expected size. It tells you if the PCR reagents are working, if the primers are working, if the PCR program is correct. Now if by chance the positive control fails to produce a band, you cannot trust the experiment. Now comes the no template control. It contains your master mix like primers, polymers, water, but it should not contain your gene of interest or your DNA at all. As a result, it won't produce any band. It checks whether your reagents are actually contaminated with DNA. For example, if by any chance the non-template control shows a band, it suggests that there might be some DNA contamination in water or in primers or maybe some contamination during pipetting. So this non-template control is merely a reagent contamination control. And then there is another control that is negative control. In negative control, it often contains DNA like your empty vector or let's say untransformed bacteria, but it should not contain the insert. As a result, it won't produce band of the target size. Now let's interpret the colony lengths. Now if you get a single band at the expected size, as you can see here, in colony 1, 5, 8, that's likely a positive colony. Now if you don't get any band, as you see in colony 2, 6, 9, 10, 12, then it probably means that the colony does not contain the target insert. Now if you get a much smaller band, as you can see in lane number 3, then that may indicate an empty vector. Now if you get a larger than expected band, as you see in lane number 4, then there is possibly a non-specific amplification or an unexpected insert. Multiple bands, as you see in lane number 7, then that's also a sign of non-specific amplification and should be treated cautiously. So which colony should you choose? Well, only the colonies that show a single clean band at the expected size. These are your best candidates for downstream confirmation by plasmid isolation, restriction digestion, or sequencing. And that's colony PCR, a simple but very powerful way to quickly find the right clone before investing time in further analysis. However, some labs they do not add the colony directly to the PCR mix. Instead, they lyse the colony and use the supernatant as the template. Basically, they pick a very small amount of colony and then they suspend that colony in the sterile water or tea buffer. Then they heat it for around 95 to 98 degrees centigrade for around 5 to 10 minutes. Then they vortex it, then they centrifuge, and finally they use the supernatant as the template and they add it to the PCR mix.